Hi, and welcome to this next video on OCR A-Level Biology. We're now starting Module 2.4, which is all about enzymes. And this is just an introductory video to what enzymes are and how they work. So enzymes, these are known as biological. So enzymes are basically biological catalysts. So what are catalysts? Let's zoom in here and see for ourselves. Let me recenter. So, what are biological catalysts? So, a catalyst is anything that speeds up, is a substance that speeds up or increases the rate of a reaction. So, enzymes essentially increase the rate of reaction and they are not used up. So, they are not used up. So, here we have an enzyme, which I will draw now. So here, this is an enzyme. And these are, I like to think of enzymes as like Pac-Man. So if you see what Pac-Man looks like, so this is a Pac-Man or a relative version of a Pac-Man. You can see that enzymes and Pac-Man are really similar in a sense. So enzymes. So why are enzymes so remarkable? Well, chemical catalysts are usually need very high temperatures and they have also need increased pressures and extremes of pH. Enzymes speed up often at neutral pH, so enzymes work best at neutral pH and also at normal pressures. So they often work at neutral pH, at normal pressures, and they're able to function at lower temperatures. Like the body temperature of 37 degrees Celsius, they're able to work at that temperature. Which is why they're very useful in biology to sustain life. So they're also very specific. You have a specific enzyme for a specific type of reaction. For example, if you're trying to digest lipids into or triglycerides into fatty acids and glycerol, you will need a specific enzyme to digest those lipids, and that specific enzyme is lipase. Likewise, you need a specific enzyme to digest proteins, which is pepsin or trypsin, and you need a specific enzyme to digest carbohydrates, which is amylase. So enzymes are very specific. So the structure of an enzyme determines what function. Because they're so specific, the structure of the enzyme determines what the substrate and is. So for example, if we take this enzyme from the top here, if I can move this up, there we go. So if we take this enzyme here, and then move this one to here. So we take this enzyme, we can draw the specific substrate for that. And the specific substrate that will fit in here is this. So the enzyme will work on the substrate and either in order to turn it or to convert it into its product. And this is what we call an enzyme substrate complex, or an ESC. So an enzyme substrate complex is formed when an enzyme joins or binds with a substrate at the active site. So the active site is the active part of the enzyme and it's where the substrate actually binds. So in this example, this little section here is the active site. And different enzymes can have different active sites depending on their substrate. So this is what I was talking about, where the structure determines the function of the enzyme. Because if an enzyme that catalyzes a metabolic reaction, a certain metabolic reaction, has a mutation and so has a change in that structure, then the enzyme might not be able to bind properly to the substrate and that metabolic pathway cannot occur really well. Hence, a metabolic disorder might result. So, as I was talking about the active site, let's move into what the active site is. So, the active site is crucial because its shape is complementary, has a shape which is complementary to the substrate. Each type of enzyme is highly specific in its function, and it can only catalyze a reaction involving the particular type of substrate molecule that fits into the active site. So that's what we mean by complementary. It can only bind to a specific substrate. And it can be altered, and the shape of this active site can be altered by a number of different things. For instance, can be altered by pH, and we'll talk about these more in a separate video because there are actually lab-based or practical-based questions that can come in the exam about this, about the pH, what else, temperature as well. So these will come in a separate video, in a separate playlist about practicals, because these are practical-based questions that you need to know. So where enzymes work? So enzymes work either, keep forgetting that this is not one note. So location, where enzymes work. So enzymes can either work intracellular or extracellular. So you have intracellular enzymes, which basically means that they work inside the cell, and they can catalyze many metabolic pathways. And they are used in various metabolic pathways. For instance, glycolysis, there's many enzymes involved in glycolysis. So involved in metabolic pathways. And in those metabolic pathways, the reactants / intermediates are the substrates for the enzymes. So there's a good example on in your textbook. There's a good example on page 101 of the textbook, but I might draw up my own, just because why not? So let's say, actually I might use the example of glycolysis. You'll learn glycolysis in year 2, but I'm just going to use it as an example. So in glycolysis, we convert glucose into pyruvate. Now I'm going to do the first half of a small section of this pathway. You'll learn more about glycolysis in year 2. So we have our molecule, in this case glucose, that gets converted into glucose 6-phosphate or something called G6P, and this is done by one enzyme, and that enzyme is hexokinase. So in this example, the glucose is our substrate 1, and the hexokinase was our enzyme A. And then our G6P is now substrate 2. The next step is that this G6P gets converted into something called F6P, and I'm using the wrong color, into something called fructose 6-phosphate or F6P. Again, you'll learn about this more in year 2. And the enzyme for this is phosphoglucose isomerase. So this is our enzyme B, which is this phosphoglucose isomerase, converted our G6P or our substrate 2 into substrate number 3. And then this fructose 6-phosphate then gets converted into something called fructose 1,6-bisphosphate, and that is done by the enzyme, can I remember the name? Phosphofructokinase, or by the enzyme PFK. So PFK is our enzyme C, and enzyme C has converted substrate 3 into a final product 4. Well, it's not a final product of glycolysis, but in this stage of glycolysis, it is the final product. Fructose 1,6-bisphosphate. So you can see here that enzyme A converts substrate 1 into substrate 2. Enzyme B converts substrate 2 into substrate 3. Enzyme C converts 3 to 4, and so on, until you get the product, which is pyruvate. But we're not going to talk about that right now. So yeah, this is an example of a metabolic pathway that enzymes control within it, that enzymes are involved. Okay, so this is an example of where enzymes are involved in a metabolic pathway within the cell. So all of these enzymes here, these are known as intracellular enzymes. Okay, still with me? Good, you're still with me. Now we also have extracellular enzymes. Actually, why am I writing that? No. We also have extracellular enzymes. Now, extracellular, these work outside the cell. Now, these work outside the cell, and they act on their substrate extracellularly. A great example of this is fungi, such as the bread mold, which releases hydrolytic enzymes from their cells from the flagellate hyphae. Those enzymes are secreted from the hyphae, and those enzymes digest the carbohydrates, proteins, and the fats found in the bread. And the product of those digestion, glucose, amino acids, glycerol, and fatty acids, these are absorbed into the fungal hyphae for use in respiration and growth. So there's an example of extracellular enzymes interaction with fungi. Also, there's also extracellular enzymes in the human body. For instance, amylase. So amylase converts a starch or carbohydrates into glucose, and this is an extracellular enzyme. It's secreted from our salivary glands in saliva and acts on its substrate outside the cell. So that's amylase, which is another example. Another example is also trypsin, which is made in the pancreas, but again secreted extracellularly at an extracellular level to help digest proteins in the small intestine. So that is the end of this video here. I know this video is quite short, but this is just an introductory video to enzymes and what we're going to be covering in this module 2.4. Thank you very much for watching. Hope this was useful, and I'll see you in the next video. Goodbye.